Okay, so we are in the last day of the conference, so as you can see, there is no longer this feeling of excitement, but I'm happy to see all of you. I actually know everybody here in the room, so it's nice, it's fun to have everybody here. Thank you for coming, and I do hope that it will, I'm sure actually, that you will enjoy this session, because I know I'm waiting to hear all the lectures that we will hear today. And we want to start with Tamar Keasar from Haifa University, and yeah, please.
Hi, good morning. Thanks for coming. So I will be talking about interactive effects of climate and land use in plant-herbivore-parasitoid communities. And I want to start by introducing the climate part. So we all know that we are in an era of climate change, and part of this climate change, at least in our region, is drying. So the forecast for the next 30 or 40 years in the Middle East is a decline in rainfall that would be between 10 and 20% of the annual precipitation. And of course, ecologists wonder what this would do to food webs and to multi-trophic interactions, and there's quite a lot of work done on this question. And the bottom line is that the effects are mostly negative. So if we consider a food web that has 3 trophic levels, it has plants, herbivores, and carnivores. This would be the situation with abundant rainfall, but if there's little rainfall, then there's little water, less water to support the plants, less plant biomass to support the herbivores, and less herbivore biomass to support the carnivores. So everything is expected to decline in abundance. So this is already old news. I'm not going to talk about this. What I do want to talk about is how this phenomenon would interact with land use change. And the reason that this question is interesting is that dryland agriculture is expanding. There's more and more drylands used for agriculture, both in Israel and globally. Here you can see the different types of crops that are grown in drylands all over the world. And actually, the question that I want to discuss today is would or do the negative effects of aridity interact in any way with land use? And when I say land use, I'm going to compare croplands with natural habitats. And this question was studied. It was mainly the work of Miriam Kishinevsky, who was a PhD student in my lab and is now at the University of Wisconsin. And when exploring this question, we raised 3 hypotheses. The first hypothesis is that there is no interaction between rainfall and land use. So in that case, if we look at increase in rainfall, we would expect an increase in abundance of plants, of herbivores, and in carnivores. And this increase would be manifested in the natural habitats, which is the brown line here, as well as in agricultural habitats, which is the blue line here. And the effect would be similar, regardless of the type of land use. The second hypothesis that we considered was that agricultural irrigation would mitigate the effects of aridity. So this hypothesis considers the fact that much of dryland agriculture is irrigated. Here we can see the extent of irrigated agriculture in Israel, all those bluish and reddish dots. And you can see that irrigation extends far down into desert areas. And irrigation can be either through sprinklers or drip irrigation. But one way or another, if we're thinking about drought as stress, then irrigation might reduce the stress for crop plants. And for this reason, we might expect the situation in croplands to be a bit different from the situation in natural habitats. So still, with increasing rainfall, we would expect increasing abundance of all three trophic levels, but the slope might be less steep than in this first hypothesis. The third hypothesis that we considered was that there would be an interaction between rainfall and land use, but it would be the other direction. And this hypothesis assumes that agricultural stresses exacerbate the effects of aridity. So we know that croplands are exposed to a lot of man-induced stressors. It could be spraying, it could be fertilization, it could be plowing, and so on and so forth. And this may make the world more difficult, even more difficult in dry lands than in well irrigated areas or places with a lot of rainfall. And therefore, we would expect an increase in abundance of all trophic levels in agriculture with rainfall, but this increase would be steeper than under the first hypothesis. So these are the questions that Miriam set out to investigate, and the way she looked at it was to conduct a field survey of agricultural and natural habitats. So the agricultural crops she looked at were pomegranate orchards, and she selected pomegranate orchards along Israel's rainfall gradient. So some orchards were up north and received about 800 millimeters of annual rainfall, and some orchards were not far from here. They received about 100 millimeters of rainfall. So this was the rainfall gradient. To look at land use, she chose orchards that were bordering natural habitat, at least on one side. She sampled both the orchards and the natural habitats, and she sampled 3 trophic levels in each of those sites. So the first trophic levels are the plants, and the plants that we considered were herbaceous vegetation that grew either inside of the orchards or in the nearby natural habitat. So you can see here, for example, the vegetation that grows inside the orchard, and here in a closer view. And in fact, a lot of that vegetation was what farmers consider weeds. So they're not happy to have them in the orchards, but they do have them anyway. And some of the vegetation was wild vegetation outside of the orchard. This is what it looks like in spring. In summer, of course, it's browner and drier. So we looked at species diversity and percentage cover of the vegetation. For the herbivores, we suction sampled insects from the vegetation. You can see here our suction sampler. And we collected all insects and classified them. And we found that we had 3 main groups of herbivores. We had leaf hoppers, we had white flies, and we had leaf miners. So all of them are plant-feeding insects, and several of them are also crop pests. For the carnivores, we considered parasitic wasps. So parasitic wasps are a very large group of hymenopterans. They number hundreds of thousands of species. The exact number is not known. Their immature stage is parasitic. So what you can see here is the life cycle. An adult parasitoid lays an egg within the herbivore. The egg hatches, and there's a larva that develops inside the body of the herbivore, consumes it from within, kills it, and eventually emerges as a free-living wasp. And because of this life cycle, parasitic wasps are major natural enemies of crop pests and are often used in biological control. In our surveys in agricultural habitats in Israel, we found a very large variety of species of parasitic wasps, over 300 species, and here you can see some of the beautiful diversity of this group of insects. They're very tiny. Most of them are 1 millimeter, 2 millimeters large, or even smaller. So these were our carnivores, and to sample the insects, we set up 2 transects that we sampled that went from the natural area, which is shown here, into the center of the pomegranate orchard. The other side of the of each orchard bordered another agricultural plot, which sometimes was again pomegranate, sometimes it was something else, but all of this is agricultural area. So what did we find? Let's look first at the results of our samples in natural areas. And when we looked at plant cover in the natural area, we found that, as expected, plant cover increased with rainfall, so it showed this pattern of increase. It was positively affected by annual precipitation. When we looked at the abundance of herbivores, we saw just the same pattern. So those green marks indicate where a positive relationship exists between rainfall and abundance. And when we looked at the carnivores, at the parasitoids, we also saw the same pattern. Now we are going to look at what happened inside the orchards. So when we looked at plants, we did see this pattern in the margins of the orchard, but not toward the center. When we looked at herbivores, we also saw the same. So actually, inside the agricultural area, there is some effect of rainfall, but it is weaker than in the natural area. And this is in accordance with our hypothesis 2, which assumes a lower slope of this relationship in croplands than in natural habitats. And when we looked at the third trophic levels, the parasitoid, actually we found no relationship between rainfall and parasitoid abundance, anywhere in the orchard area. So actually the slope here is kind of flat. Then we looked at parasitoid species richness and species diversity. Here we did find some relationship between rainfall and species number and diversity, but again, mostly in the margin parts of the orchards. So to conclude so far, we found the aridity effect on the abundances of plant, herbivores, and parasitoids, but they were mitigated within the pomegranate agro-ecosystem in accordance with our hypothesis 2. We found that parasitoids experience the highest mitigation, so they're less affected by climate when they sit inside the orchard. And this was quite interesting and cool, but we wanted to dig a bit deeper into that and focus on the parasitoids. And we wanted to ask what happens to the community composition of parasitoids under these conditions. So for this, we looked at the sub-sample of the parasitoids that we collected. You can see that the sub-sample was quite large. It was almost 3,000 individuals of parasitoids that we collected in those very same orchards. And those parasitoids we selected because we could classify them to species. Taxonomy is quite hard with parasitoids, but with those we could tell the species. And we had some information about life history traits of those species, so we could use them in additional analysis. What we did for this subset of species is to relate species composition to environmental conditions, and also related their life history traits to environmental conditions, trying to look for traits that might confer adaptations to specific climates or land uses. For this, we used the fourth corner and RIQ analysis as our statistical methods, and our aim was to identify traits that correlate with arid or mesic habitats and traits that correlate with natural or agricultural habitats. So this is the first part of our effort. So first we used a PCA analysis to cluster together communities of parasitoids that were similar or dissimilar in composition to each other, and we asked what explains community composition in terms of environmental conditions. And perhaps not very surprisingly, we found that climate plays a major role. So aridity explains 63% of the variation in community composition, and we could plot it on this PCA graph that basically the x-axis is a climate axis. So on the right-hand side of the axis, we see the more mesic sites. They're characterized by proximity to a stream, a high NDVI, photosynthetic activity, high richness of herbs, and high cover herbaceous vegetation. The left-hand side of the axis are the more arid sites or arid seasons. So these communities were sampled, for example, in late months, in the middle of the summer, when the weather is hotter and drier, at places with high mean temperature, and in more eastern localities, far away from the sea. So this was one important axis, and the second important axis was the axis of land use, which you can see here, and it explained about 21% of the variation in community composition. And here we identified communities that mostly lived in the orchards, up here, and communities that lived mostly in natural habitats, down here, and also communities that didn't care much about land use, that you will see in a minute. When we looked at traits that correlate with these environmental and land use variables, we could identify three groups of traits that made some sense to us. So the first group of traits that you can see here is located at the right-hand side of the x-axis, and it has to do with living in mesic environments. The second group of traits, which is up here, is correlated with agricultural environments. And the third group of traits, which is at the bottom left part, is correlated both with arid environments and natural habitats. So what were those traits that were correlated with each set of conditions? So let's first look at the arid and mesic axis. So arid conditions were correlated with parasitoids that mainly attack whiteflies, whereas parasitoids in mesic conditions tended more to parasitize plant hoppers and leaf hoppers. Parasitoids from arid sites tended to parasitize larvae of other insects, whereas those coming from mesic conditions prefer to parasitize egg hosts. The ones from the arid conditions were mostly ectoparasitoids, which means that the parasitoid develops on the outside of the host body, whereas those from the mesic conditions were endoparasitoids, so they entered the host body and fed on the host from within. And finally, those from the arid conditions were mostly coenobionts, which means that they let their host grow and develop as it is parasitized, whereas the ones from the mesic conditions were mostly idiobionts, so they stopped their host development upon parasitism. We did the same exercise for the parasitoids that lived in croplands versus natural sites, and found that those from natural habitats mostly parasitized moths and flies. They were mostly ectoparasitoids and mostly had idiobiont development, whereas those from croplands parasitized mostly exposed hosts, were endoparasitized and had coenobiont development. So according to these traits, we could classify those 27 parasitoid species that we analyzed into 3 groups. So this group is a group of climate specialists. Each dot and abbreviation here indicates one species of parasitoid. So we had about 15 species here. And these are the parasitoids that like mesic conditions, they like the wet habitats, and they don't care much about whether it is agricultural or natural habitat. The second group of parasitoids are the land use specialists. So these are the parasitoids that specialize on agriculture habitat and don't care much about climate. And the third group of parasitoids is down here, just 2 species, which are both climate and land use specialists. So they like the desert and they like the natural habitat. So to conclude, we found that plants, insect herbivores and parasitoids are more resilient to drought in irrigated orchards than in natural habitats. We identified parasitoid life history traits that correlate with life in arid environments and in croplands. And since both arid environments and croplands and arid croplands are currently increasing, we predict selection for these traits under climate change and the accompanying increase in irrigated dryland agriculture. And with that, I'd like to thank our funders and collaborators and different teams and have a training session.
Yeah, thank you, Tamar. We have plenty of time for questions, so yeah.
Thank you, Tamar. So, your hypothesis, you started with the bottom-up effect. I read a study recently in Chile where they see it a top-down effect. So is it possible that like we might see the same effect here in Israel or in the Middle East? Like the parasitoid get affected first and you don't have enough parasitoid to control the parasites.
You mean as a result of climate change?
Yes.
Yes, I think it might be the case in natural habitats, but in agricultural habitats, since we saw that irrigation can compensate for a lot of the climate change, and especially for parasitoids, I think that actually parasitoids would do better than the other trophic levels in croplands because of their higher resilience. So I think, if anything, then the top-down effects would become stronger.
Thank you so much for the talk. I might not be clever enough to get all of your wonderful graphs, but I do have to ask. I mean, we did see that in more mesic sites, we have more herbivores that can potentially be hosts for parasitoids, but there is also a difference in how easy our different species are to become parasitic of in the beginning. And it's like, that's another element that maybe I missed and you did address, because, you know, if you have aphids and they're more easy for parasitoids to become hosts of, or there is a greater variety of parasitoids supported by these, and they're in the more arid sites, then that perhaps also kind of affects why we see that mediation between sites.
Okay.
It was my question. I don't know, maybe I missed you.
Let me go back to this slide. So basically you're saying, one more. If these hosts are less vulnerable to parasitoids than these hosts,
or the other way around. I mean, if one set of hosts is more vulnerable or less vulnerable, that also kind of dictates what parasitoids can hunt on him, or the amount of species of parasitoids that might be into that specific host and stuff like that. I don't know if it was taken into consideration, or is it just like, because I just saw this and I thought I wonder if they checked aphids, which I know are more easy to parasitoid than, let's say, spider mites or something like that.
Yeah, it's a good point. The problem is that we know very little about the host range of most of the animals that we deal with. As I told you, we have more than 300 species. Most of them we couldn't even classify to species. It was just morphospecies, and we could tell their family or genus at best.
Okay.
So we don't really know what they eat.
Okay.
But it's a good point.
Okay.
Yeah.
And I have another, that's an easier question to answer, hopefully. I mean, we're looking at all of this and this mediation that you found that irrigation allows for more arid sites. And I'm wondering if there is perhaps a talk or a consideration of talking to farmers, or perhaps legislation of, like, you know, let the plants around the field just kind of live, or something like that. Was that something that was considered?
Yeah, so we've done that, not with the pomegranates, but with vineyards. And in fact, we were successful. So most of the vineyards in Israel now are letting the herbs be.
Yeah.
And it was a very interesting process, because we worked with a winery and we worked with a team of social science researchers. And we could get input from the farmers and understand what made them do this switch in management. Turns out that they didn't care at all about biodiversity or biological
It's
control.
just cheaper for them when it comes to not
It's
buying.
not, it's not cheaper. Economically, it's the same. What they do care about is soil erosion. And the minute they see that vegetation cover reduces erosion, then they're very open to change. The change also was facilitated by the market structure of this wine industry, which is a bit different than other markets, but it's doable, and I think it's something that we should aim for.
Okay, cool. Thanks.
I also have a question. Thinking about your results, it's actually, of course, surprising to me, because I would assume, and I keep on telling my students also, that the natural habitat will be more resilient. So, do you have any idea about the forces that actually, how can it be that actually the managed habitat is the one that is more resilient to, especially to water or aridity?
Yeah, I think the key here is irrigation. So the minute you irrigate the croplands, then they become richer and they become kind of a refuge. And actually, Michal did a study on vineyards here in the desert, and she found that there was a seasonal migration of parasitoids. So in the summer, they move into the vineyards. In the winter, they move out. So they follow the water, I think.
Okay, and did you check? You did check the differences in diversity between the natural habitat and to see whether it's the same species migrating or actually completely different species, maybe invasive species coming in.
So Michal did, and she found that the composition was very similar, at least here. Actually, she expected the opposite, because she expected a bigger contrast in conditions between nature and agriculture here than elsewhere, but the composition was really similar.
But for the specialized arid species, you showed just 2 species, right?
Yeah. So they won't move into the ocean.
They won't move, and they're more resilient to the natural conditions.
Yeah.
More questions? If not, then thank you very much.
Thanks.
And we apparently will do it in our own pace and not in other people's pace, if that's okay. We are moving to our second talk. Actually, we had one cancellation because Ariel is sick, so she is apologizing and she will not come to give us a talk. Then we will move directly to Sanjana Sarode.
Good morning everyone, my name is Sanjana, master students. Today I would like to explain about my master project which I entitled as Aphid-Mediating Virus Infection Alters the Volatile Organic Compound of Emission in Pepper Plant. So plant virus significantly have yield loss up to 1/3 of the economical loss worldwide, and approximately 55% of the plant viruses rely on aphid vectors for its transmission. So when the insect feeds on infected plants, it picks up the virus inside it and it becomes virulliferous, and next this virulliferous insect will move to the new host, and when the feeds on the new healthy plant, it injects the virus inside the plant cell, and this inoculation will start the infection cycle, and this is how the cycle continues, and this is how it start transmission. But how does the virus ensure that they reach to the new healthy plant? So one of the smartest strategy the virus uses is changing the host preferences. When the insect feeds on the virulliferous plant, it picks up the virus inside the plant, and once it picks up the virus, and the virus inside the aphid will redirect its preferences, and it more attracted to the healthy plant for its transmission. So what is the mechanism behind this host preference shifts? So one of the major mechanism the virus uses is changing the host physiology. So here is the evidence from the literature. The x-axis is the time which is spent, and the y-axis is the non-virulous vector. Here is the virulous vector. Here you can see the non-virulous vector always attracted to the infested plants, and here when the non-virulous vector is always attracted towards the healthy plant. Okay. Here also we see the similar pattern. And this similar what is the mechanism behind these host preference shifts? Because one of the major mechanism which come from the part is changing the volatile organic compound, and plant produces several major volatile compounds, mainly fatty acid derivatives, GLVs, terpenoids and phenylpropanoids, which also helps in pollinator attraction, abiotic stress responses, plant-plant signaling as priming, and also indirect plant defenses, and also modulating the host vector preferences. And my research is focused mainly on the indirect plant defenses and the vector manipulation. For that, we have the hypothesis, whereas when the virulliferous aphid feeds on the plant, the volatiles inside the plant is changed into virus-induced volatiles, which can change the behavior of the vector. So to support my hypothesis, we use capsicum annum as a host, and the vector is Mycus persica, and the virus we are using is pepper yellow vine virus, which is very common in the pepper plant. So we have three different treatments: control, which is very healthy plant, and the Mycus persica, which is inoculated which is a non-virulous aphid, and we have virus aphid, which is inside the pepper yellow vine virus. And this experiment is conducted by Galina from Volcani Institute, and we collected the samples. After the infestation period, we extracted the volatiles by using solid phase micro extraction and analyzed using GCMS. The first question we asked is whether these treatments have any effect on the volatile profile. So the PCA1 explains 41% of variance. The PCA2 explains 30% of variance. So clearly, overall volatile profile, it shows clear separation between all the three treatments. And we also have common and unique volatile compounds among the treatments. We have 35 volatile compounds which are common in all the three treatments, and we have 50, 29 and 28 unique compounds in control, non-viruliferous aphid and viruliferous aphid respectively. When we see the heat maps, we can see among all the treatments there is a differential emission patterns in the volatiles. Also in the class level, we can also see differential emission pattern. So we wanted to see how these volatile compounds behave at the individual compound level. At the individual compound level, so first we would like to see the fatty acids and GLVs. If you look at the non-virulous aphid, there is no significant changes when you compare it with the control. But when there is a virus infection, most of the volatile relative concentration has been increased. So which suggests that under viral infection, these volatile compounds which are related to fatty acid and GLVs has supported aphid attraction and transmission. And the next important class in the plant defenses is terpenoids. As I expected, there was no similar pattern in the terpenoid classes, but in the linalool, we can see there is a increase in the concentration at the non-virulus when you compare it with the control. But when there is an aphid infestation, this has been suppressed. So which might be the virus is suppressing the defense-related terpenoids for their own use, which can help to attract more aphids. And in case of these volatiles, there is no differences between the non-virulus and the virulus. But if you compare the non-virulus with the control, there is a separation in the concentration of these volatiles. So this may be due to the aphid itself showing their defense mechanism to the plant for their own use. So here we saw both from the aphid defenses to the plant, and another important class for the signaling and also the chemical and physical barriers are benzonoids and phenylpropanoid pathways. So here, if you look at the non-virulus concentration, non-virulus treatment, there is a significant decrease in most of the volatiles, and in the virus infection, all these volatile compounds has been significantly increases, which supports the attraction of the vector towards the plant. But if you see here, there is also increase in the concentration in the non-virulus when you compare it with the control, which also simultaneously modulate this plant signaling to the other plants. To conclude all, first we saw there is differences between all the treatments, and in the GLV and fatty acid derivative classes, we saw the elevated volatile concentration in the virus infection, which enhanced the aphid landing and host attraction cues. And in the terpenoids, we saw the reduction in the virus infection due to which may helps in the weakening the plant defenses and reduce the plant deterioration. And in the benzonoids and phenylpropanoid concentration, we saw there is an alteration in the volatile profile, which enhance the vector attraction and also giving the signaling to other plant within and other plants. In summary, virus induced changes to the plant volatiles, which amplify the attraction towards the aphid and also for helping the virus dissemination. So to come, I would like to thank my professors Verid, Merav and Oz for supporting, and Galina for conducting the experiment, and my friends and labmates for supporting, and funding agency for funding my project. And thank you for listening.
So thank you, Sanjana. Plenty of time for questions.
Thank you. How does the virus do it?
Sorry?
How does the virus do it? What is the pathway that makes the virus induce the plant to change its volatile profile?
Yeah, the virus will manipulate the biosynthesis pathways of the volatiles, which are giving the defense mechanism for them, which are not, which deter the aphid, and the virus trying to reduce those genes related to those biosynthesis biological processes for their own benefit for the transmission.
But do you know how? Do you know
the
anybody
biochemistry?
knows what's the real mechanism biochemically? Do they excrete something in their saliva, or do they? No, it's not. I'm talking about the aphid now. Sorry, it's a virus. Do they know how what they change in the?
They change
DNA?
the gene. They change the genes. So we are also trying to look at the genes related all these changes in the volatile.
It's a retrotransposon.
Sorry.
I'm sorry. Because if it changes the gene itself. Okay.
In the bio,
the expression.
Yeah,
okay,
in the
okay,
expression.
okay.
Yeah.
But,
sorry, that's on me.
Yeah.
but this is something that you try to do and didn't work. Yeah.
Did you try to infect the plants by the virus itself without that?
No.
Is it possible?
It's a nice control.
Yeah.
This would be a nice control.
Yeah, but no, we use the virophorus vector for the infestation.
Yeah, it's a nice. I think it's an important comment that it should have been. It's a good control.
Yeah, it's a very.
They do it with fungi a lot. That they just they take a leaf that has been infected, they make a few holes in the plant that hasn't been infected, and they just kind of rub the juices from the leaf of an infected plant on it, and that kind of infects the plant.
Yeah.
It depends the mode of transmission.
It depends upon.
It's a circulative one or?
Yes, non-circulative persistent.
Non-circulative.
Yeah.
Okay.
More questions? Although we had now a full discussion, but okay. So thank you very much, Sanjana. And we are continuing to our session with Barazani from Volcani.
Number of things. מרב, זה בסדר, אני אשב?
בוודאי.
כן.
בוודאי. אתה צריך רמקול או שאתה בסדר ככה? רגע, חכה, זה בסדר שנסדר את זה ככה ש...
אבל אני רוצה לראות.
אתה רוצה לראות?
אני אשב ככה.
בשביל מה אתה צריך לראות? רגע, אני אביא לך עוד.
זה עם הפוינטר.
כן, אני אביא לך, זהו.
אבל כן, אני קצת קוראים לי.
רגע, אני אזיז. אתה רוצה לי?
נצטרך על המסורת
מסתכלים על
כאן בסדר?
כן, מצוין. תודה רבה. So thank you, Merav, for inviting me and letting me introduce the work we are doing for many years, studying the Eruca sativa in the lab. It's one piece of a big, 1,000 pieces puzzle that we are trying to understand genetic variation, intraspecific variation, genetic variation in Eruca sativa. And here I will talk about the interaction of the plant with the herbivores and pollinators, and we are trying to understand the adaptation of a desert population to a specialist herbivore named Plutella xylostera, which this is diamond backmoth, DBM. So Eruca sativa, you probably know it as a salad rocket or Arugula. It is known as a salad herb. It's a winter annual, self-incompatible, relies on insect pollination, and in Israel it grows naturally along the Jordan Valley, but also in the northwestern Negev. In the Jordan Valley, it grows from desert environments in the south part of the desert valley, of the Jordan Valley, not far from the Dead Sea. And then along the way, there are populations scattered, and the northern distribution range is on the east coast of the Kinneret. So we study desert versus Mediterranean habitats, and although there are the geographical distance is not that big, I mean it's 70, 80 kilometers with several populations scattered in between. There is a possibility of gene flow. Yeah, there is a distinct genetic diversity of differentiation between the desert and the Mediterranean populations, and this is the focus of our study. We are trying to understand the genetic diversity. So the two sides, the Mediterranean versus the desert, as you probably guessed, are very distinct in climatic conditions, and you know the effect, and we heard from Tamar the effect of climatic conditions on the different biotic environment, but the populations also are exposed to different edaphic conditions and abiotic environment, and among others, there is also biotic interactions. In the Mediterranean plant communities, the plants are growing in a very heterogeneous, rich annual plant community, where Eruca sativa, you can see it here, is a very minor component of the annual plant communities, versus the desert, in which the plants grow in almost complete monospecific habitats, with only Eruca sativa growing there, which very much resemble an agricultural plot, right? It's just Eruca sativa growing there. Along the way, we observed a few phenotypic differences between the populations. They differ in phenological parameters. The desert plant flower much earlier and produce less vegetative biomass in comparison to the Mediterranean one, which makes sense, right? Plants in the desert are fast induced flowering to survive, to make the next generation. This is an annual plant. But very surprisingly, it seems that the desert plants prolong the flowering stage, the reproduction stage. In addition, you can see that the petal colors are completely different, and another very big difference between them is the trichomes that covers the stems of the stems and leaves of the desert plants, whereas the ones from the Mediterranean are completely glabrous, you see. They are naked from trichomes. And this also makes sense, because in the desert they have to protect themselves against radiation, and these trichomes, among others, protect the plants from radiation, right? Yeah. But they also have something to do with herbivory, right? We know about it. So before I will go into herbivory, I will just mention that we use the PST-FST comparison, which make in this comparison, what we do is test genetic diversity parameters, genetic differences, distances. This is the FST versus quantitative measurable phenotypic traits, and we found that the PST values are higher than the FST, the genetic distances, which means that these are, we can conclude that the phenotypic differences are due to divergent selection. If it would be the opposite and the FST values would be higher, then we can consider the phenotypic variation as a cause of genetic drift. Yeah, this was done, of course, in a common garden experiments. Along the way, we also identify SNP loci that have a strong association to environmental parameters, so we can conclude that the genetic diversity that we find is due to cause ecotypic differentiation between the populations. And this is how ecologists should conclude about ecotypic variation and not as commonly used to look at any genetic variation as ecotypic variation. So one thing that we also looked is we performed a very basic, not professional entomological study, and we found that the Mediterranean populations are more exposed to generalist herbivores, Lepidopteran and aphids, of course, whereas in the desert environment, the diamondback moth is more abundant, in very high density. And this was done by spreading or scattering pheromone laws in the populations over 2 years, and you can see the dramatic decrease between years. This is because of rainfall and low population size, but always that populations of this specialist insect, specialist moth, is higher in the desert habitat, which also makes sense because it's a monospecific habitat, and specialists prosper in such plant community. When testing the resistance of the plant to insect, we use no choice feeding assay with larvae of Piris, of Plitella salustella, DBM, and of Spodoptera littoralis, is a model plant of generalist herbivore. And we tested, measured the larval weight along the experiment. This is the final weight after 10 days. What you can see is that the basal resistant of the Mediterranean plant, as long as the weight is lower, it means that the plants are more resistant. The Mediterranean plants seem to be more resistant than the desert plant, but when you induce the defense mechanism in the desert plant, you see a drop decrease, which means that these are defenses in the desert plants are directly induced defenses. Yeah. So when you induce the plant prior to the feeding experiment, the defense level increased, and then the plant becomes more resistant to the plant. And this is due to what you heard before. The herbivore induces the defense system within the plant. Then there are signaling pathway, mainly of jasmonic acid, that is increased by either mechanical damage or elicitors that are secreted via the saliva, as Merav mentioned, and then it induces chemical defenses and structural defenses. Structural defenses are like trichomes, and then chemical defenses are, here I put the glucosinolate, the molecule of glucosinolate, which is the main chemical defenses in the Brassicaceae family, and the sativa is a member of this family. When testing the specialist insects, you see that the basal and defense levels are the same, and this makes sense because VBM co-evolved with the Brassicaceae family, and they know how to detoxify. They evolved mechanism to detoxify the chemical defenses, and we'll come back to this later on. So glucosinolates are the main defense compounds in Brassicaceae family. These are the intact form of the glucosinolates have no biological activity, but when the plant is damaged, this enzyme, myrosinase, is released, it hydrolyzes the glucose part of the molecule, and then a bunch of diverse chemicals start to release in the intact leaf, and which are toxic mainly to generalist herbivores, right? And the most toxic form is the isothiocyanate or ITCs, right? So when we measure the glucosinolates, the intact glucosinolates level in the plant, we see no effect of defense elicitation with herbivores on the level of one in the desert plant, but we see a significant effect in the Mediterranean, right? And there is no difference between the two populations. These are slides that summarize the results of an RNA-seq analysis, which in total, and we use RNA-seq in different, the effect of herbivore, different herbivores, and wounding and jasmine treatment on the expression level of the transcriptome, and then what in the big, enormous data that we received, we can summarize and see the differences between the desert plant and the Mediterranean plants in the signal, in response to herbivory. And what we found is, first, very interestingly, we saw that the herbivores induce different signaling pathways in the plant. The salicylic acid in the desert plant, jasmonic acid in the Mediterranean plants, and these elicit a different cascade of genes that are involved in mainly sclerophyllin, which means making the leaf more tough to eat and to consume by herbivores, whereas in the Mediterranean plants we see mainly a chemical warfare, which means that we see upregulation of the fatty glucosinolate and the release of ITCs, which are toxic to the herbivore. Here we can see also differences in genes that were linked to responsible for proteinase inhibitor and for regulation of glucosinolate breakdown. So when testing a proteinase inhibitor, we see after herbivore with these two generalist and specialist herbivore, here we use species, we see a drastic increase in the desert plant in comparison to the Mediterranean plant. If this is this graph we saw before, showing the decrease in the increase in the resistance in response to when a generalist herbivore feed on the plant. And now we can understand this increased resistance, which is due to proteinase inhibitors. When testing the isothiocyanate level that are released in response to herbivore, we see that the levels of ITCs in the Mediterranean plants are much higher than the ones in the desert plant. This was done by collecting volatiles, because ITCs are volatile compounds, using a headspace and analyzing the volatiles released from the leaves, and we see that there is much difference between the plants. So plants of the Mediterranean produce much more isothiocyanate than the desert plants. In the desert plants, we see increase in genes that are responsible for diverging the breakdown products from isothiocyanates to simple nitrates, which are less toxic to herbivores. We see a big increase in the nitrate specifier, which means that the plants of the desert plant diverge the breakdown products from isothiocyanates to nitrates. So why did they do it? One thing that we have to mention here is that ITCs, the release of ITCs attracts the specialist insects to lay their eggs on the host plants. So they use these chemicals as a cue to detect the host plant. And when testing the effect of these volatiles on larvae, we saw that larvae, freshly hatched larvae of DBM, very much prefer to feed on Mediterranean plants over the desert plants. We place the larvae on a petri dish and give them the choice to choose to feed on pieces of disc, pieces of the two leaves of leaves of the two populations, and they prefer the Mediterranean over the desert plants. We manage also to separate males from females by dimorphism. This is a male. Very clearly, you see that it's different from the females. And we used oviposition preferences using this apparatus, homemade apparatus, where we released one mated female in a big chamber and let them choose to lay their eggs on plants of the two populations. And we see again that the females prefer to lay their eggs on Mediterranean plants over the desert plants. Right? So we can conclude and say that there is probably a chemical cue, which is the ITCs, or a deterrent compound in the desert plants that either attract the female or deter them or repel them. And by making the plant less apparent to this specialist modes, the plants in the desert evolved a mechanism to resist either oviposition or repel the larvae, which is probably the efficient mechanism for herbivory of this insect. So this is the first adaptation. But then we saw also that this should have been See? No. What did we do? Because it's, can you, it's a video. So, we saw the DBM also forage for nectar, and we observed that the insect, the adults, forage for nectar on the plants. There is this paper by Merav and Foster that showed that the quantity of DBM increases after feeding with sugars. So we thought and hypothesized that this insect also is a friend. I mean, it has some mutualistic interaction with the plant because it's the main insect in this population, whether it has something to do also with pollination. So we might move forward and test this as well.
Now it works.
Yeah. And then we use the same apparatus, only this time we placed flower stalks with equal number of flowers in each side chambers, and released either male or females into the main tank, and saw the preference for foraging. And we see that both males and females prefer the desert plants over the Mediterranean plants. So there is probably a chemical cue that attracts them, because the visible clues are very limited using this apparatus. And here is the results of the chemical analysis. We found, like the work that was presented before me, a very diverse range of chemicals, but the more apparent and striking differences were found in benzoate group of chemicals, which all belong to the same biosynthetic. Oops, sorry. I jumped too far. Oh, I'm going to the other direction. We'll get to it. So you saw and
Don't remember.
they all belong to the shikimate pathway, and here they are organized in a chronological stage from the acetaldehyde, which is in upstream to the pathway, to the end product Volatile. We saw them in the previous lecture as well, these chemicals. And we see these chemicals that are very distinct in the two populations, with methyl benzoate as exclusively emitted from the Mediterranean plants during the day, and Volatile, the end product, which is exclusively emitted from the desert plant, but during the evening. This work was done here in the Bouquet together with the VED. And we see also some other compounds like glycol and benzoate, which are higher in the desert plant during the day as well. I will come back to it later on. This was all done also with the headspace analysis. And Volatile is known in other species to attract moth. Right, methyl benzoate, benzaldehyde are more serve as a chemical use to attract honeybees or other generalist pollinators. Okay. We also use the EAG system to test for physiological response to the antenna of the insect. This was done by a very talented student, Ben Peretz, who managed to excise the very delicate antenna, to attach it to 2 electrodes and test the response, the electrical response to floral and leaf veg volatiles of the two population. These are the results, summary of the results, and Shai will probably can explain more how they did the statistical analysis, but in general what we see is the effect, the different effects of flower and leaf on the antenna response, the differences in desert and Mediterranean, and in some chemicals we saw, some floral volatiles, we saw also differences between across sexes. We use mated females, males, and virgin females in this analysis. So, but so far we cannot say which compound attracts or deter the insect, just general response of the antenna. And Ben is working on that now. So to summarize the results, this schematic drawing gives us what we understand or hypothesize that happens, assume that happens. The desert plants which are adapted to DBM release different volatiles to maximize the fitness in the desert habitat. So they avoid releasing ITCs, which attracts the insect, but probably, most probably secrete other chemicals that deter the females and the larvae itself. We recently Ben found that the desert plant produce much higher concentration of this compound, which is acetophenone, and acetophenone is known to attract parasitoids. So we assume that maybe one other adaptation is the release of chemicals that attract pre-trophic levels, and from flowers they release veratrol, which attracts the DBM during the evening, but also during the day, other chemicals that attract generalist pollinators like honeybees or others to increase the possibility of pollination during the day as well. And this is it. There are different students that were involved in this project. Sharon Shafir, which is accompanying us for many, many years. Veretzin from here that was joined us when we started to analyze the volatiles, and Tamar from Volcani Center, which now try to understand the regulation of the volatile emission from the two ecotypes. And thank you.
Yeah, so thank you very much, Oz. I think that at least at some point you raised excitement in the audience because we found, we also find something similar somewhere else. So it's interesting to see how many connections, even if you are asking completely different questions, eventually plants are actually responding similarly. But yes, it's open for questions. People are fixed on coffee, I think.
I have a question.
Yeah.
Did you try crossing the two populations?
This is a big question, big things we are struggling with, because crossing the population, we tried it a few times, but it never succeeded. They don't acclimate. to the different habitats. So we failed to do it. So we use common garden experiments in Volcani Center, either in the greenhouse or in the outside, the field experiment. And we found interesting things. I skipped it, I have it below if you want to see it, but we see, for example, that the desert plant have much higher fitness output when growing in Mediterranean habitat.
Really?
This is, yeah.
Now we should talk.
I have to show you this. So what we did is we grew them outside in a common garden experiment. We tested maternal fitness and paternal fitness. And paternal fitness, what we did is we developed, based on the genome sequencing, we developed loci, markers that we can say whether in this loci plants are homozygotes or heterozygotes, and then the marker differentiate between the desert and the Mediterranean. So we see that the fitness output of number of seeds and the number of fruits per plant is much higher, significantly higher in the desert plants versus the Mediterranean plants, but also on the paternal side. So the insect collect pollen, transfer to that, and then we can test the seed itself, the seedling, and tell who was the source of the pollen. And we see that the paternal fitness of the cream offspring are much higher than the Mediterranean. So it means that the insect tend to visit more the
desert variety.
The cream of the desert plants. And this was done in Volcani, of course. But then also the desert plants contain much higher nectar volume than the Mediterranean ones. And this is also with the sugar content. And this was done in several time points during the day. So it also rewards the insects. So it's the volatiles, the color, and the reward. And then during the day and during the evening, insects tend to visit more the desert plant. This is how the adaptation to the desert environment, plants are more adapted. So if thinking about climate change, maybe it's a good thing
that was kind
for
of
plants.
the question that I wanted to ask. There is another student in my lab that did an experiment on plant volatiles and pollinator response to mustard that is cultivated mustard versus wild type mustard. And he found, rather unsurprisingly, that the wild type had much more attraction to different types of pollinators. And I'm kind of looking at this and I'm thinking, what happens if we take the cultivar and we take the desert variety? What would be the consequences? Because when it comes to pollinators, the answer is quite clear. It would probably be more attractive to pollinators. But would it make as much plant mass? Would it taste preserve itself? Would it be as uniform as cultivars need to be? Like this was something you thought about? I don't know.
I can tell you that we didn't test the cultivar, the variety, cultivated variety. But I can tell you that these plants are more pungent.
Not terribly surprising,
So I don't know what the differences is between the cultivated variety to these, but just by taste. I mean, you cannot put them in the salad.
I imagine it's usually what you get.
But we do use the cultivated variety in pollination studies, and we see that it's highly attractive to pollinators.
It's highly attractive.
Very much.
Yeah, I mean, you enter the greenhouse during the growth phase and you smell them. It's wonderful. I mean, the first, when approaching a garden and telling her about this, this is because we smell the differences between the two ecotypes. You smell it. So if you smell it, you probably will detect it. So yeah, we use the expertise of that to test the volatile differences.
Which the one question I have to ask: are they the same species actually?
Yeah, and they cross and they make.
Yeah, they cross.
Yeah,
okay.
They are the same species.
Because maybe it's on the verge of. It's actually I
yeah,
think
but
this
with
speciation.
brassicas it's very. In this family, it's very, very. You can cross even two genera. Sometimes the genetic limits there, but these two populations cross and they are the same species.
Yeah.
I mean, and they cross and they produce fertile offspring.
Cool. So really, thank you. Thank you to all the speakers. Coffee time. We are back here in 1:45 for another exciting session for me.
אני אלך ואחזור, אני חושבת, אני רוצה לראות מה קורה עם
נכון?
כן, דברים טובים, אני לא יודעת. שאלתי שאלות.
אני מזמינה אנשים שאני רוצה לשמוע. כן, תודה רבה.